Yay, okay. So today we're going to be talking about bacteria. If you recall, I'm going to use the annotation tool already, and I'm going to be a cool kid and make lots of annotations. Just give me a second to... All right. Okay, so as you guys remember from last class we sort of took a step back and talked about the 3 domains. And so those were Archaea, Eukarya, and Bacteria. So a domain is the largest classification of life. So all living organisms on Earth can be classified into either Archaea, Eukarya, or Bacteria. So today we're going to be looking at domain Bacteria. And so if you had to describe these organisms in 2 words, it would be unicellular, which means one cell, and prokaryotes, which means that there are no membrane-bound organelles. So this just means that they lack compartmentalization of their inners in their cells, so they can't be very complex. Now, something you need to keep in mind, these 2 traits also pertain to Archaea. So that's when we have to look at further differences on the molecular and cellular level to differentiate between them. But for now, just think of bacteria as unicellular prokaryotes. So something that all prokaryotic cells have in common is that they're very small. So a micrometer, or the symbol you might see, prokaryotes have cells that are 0.5 to 5 micrometers. So because bacteria are, again, unicellular, that means that their entire body is just 0.5 to 5 micrometers. Now, compared to eukaryotes in domain Eukarya, they have cell diameters that are around 10 to 100, and they're also made out of multiple cells a lot of the time. So they're going to be a lot bigger, which is why we can see other like rabbits and cats, but we don't really see a lot of bacteria with the naked eye. So now moving on to distinctions between Archaea and Bacteria. Wait, can you guys see my pointer thing? Like the pen moving around, or can you just only see my annotations? Okay, I'm gonna assume that you guys can't see my pointer thing. But anyway, so differences between Archaea and Bacteria. Oh, you guys can see it? Okay. So even though they're both unicellular prokaryotes, and these prokaryotes both have cell walls, one difference is that the cell walls of bacteria are made out of this special thing called peptidoglycan, and it's a sugar that is unique to bacteria. And so one way that antibiotics are made are that they often target peptidoglycan specifically in the body, so that the cell wall of the bacteria breaks down and the cell dies, but then it also doesn't hurt humans because we don't have cell walls and our cells don't have peptidoglycan. So it's a good way of differentiating between bacteria and other organisms. So they also have something called capsules on their outer layer, which is outside of the cell wall. So don't think of the cell wall as the most outermost layer. Sometimes there's more. It's a sticky layer of carbohydrates, which are not the same as like rice and bread carbs, but carbohydrates, sugars, or protein. If it is dense and well-defined, so that means that it is like compact, it is strong, then that means it's a capsule. But then if it's not either of those things, it's called a slime layer, but they essentially have the same function. So it helps individual bacteria stick together in a colony, and so this colony can protect against dehydration, shield pathogens from attacking bacteria. And so when they form a colony, an example of this would be something called a biofilm, which is like a bunch of bacteria stuck together. In our guts, there are a lot of biofilms on our teeth, like the white stuff, that's a biofilm. And so it just basically makes it stronger, makes like a protective covering, yeah. So another aspect of bacteria structure is called Fimbriae, which are hair-like structures, appendages with, again, sticky uses. So that's going to be a recurring theme. They are shorter and more numerous than something called pili. So pili we will look at later when we're talking about bacteria reproduction, mostly used for transferring genes along one another. Probably have seen this word a lot, flagella. It's like you have a cell and then you have your little thing. This is probably what you imagine sperm to look like or something. This is the flagella. It moves. It helps it move. It's thin. It has no membrane. And there are three parts of it: motor, hook, filament. And so scientists believe it to have evolved from ancestral pili, again, the pilus, and it just like grew longer and helped it move over time. So looking at the diagram on the right, this would be the flagella capsule. It's outside of the membrane cell. So this, we can also add like another layer and call it the cell wall. And yeah. How do I clear all drawings? Okay. Okay, so the genetic material of bacteria. So because bacteria size and complexity is the main thing, oh, sorry, that's not undo. It's less than that of eukaryotes. It's going to have less DNA. So that means that there's less proteins to code for and less things that it needs to keep it going, which makes sense if you think about it, because if a eukaryotic cell is so complex and like has all of these different compartments and different organelles carry out different mechanisms and metabolic processes, then it's going to have more DNA and more instructions. So that's why prokaryotes generally have less DNA. And so if you guys have ever seen a karyotype, which is like a map of all of our chromosomes, which contain all of our genes, it looks like this, like kind of like this. You have like pairs of chromosomes. So this is linear, it goes up and down. But then the genome of a bacteria is actually circular. And so these circular chromosomes are located in the nucleoid, which appears lighter than the surroundings in electron micrographs, which are a way of visualizing. So they may also contain independently replicating DNA molecules called plasmids that are also circular. And something you should know about plasmids are that they are very useful in DNA genetic engineering, because you can like say this is a plasmid and on this plasmid you have this gene. It's really easy to just like, obviously not with scissors, but I'm just saying, to cut out this piece of gene and like insert it somewhere else. Like it's really easy to do that with plasmids. Sorry, I'm just revamping my DNA model, but yeah, that doesn't look right. Anyway, so yeah, in an electron micrograph, this part would appear lighter. Again, the bacteria is like circular, it's connected, it's not just linear. And then plasmids are different from the nucleoid region. Okay, so how do okay. Things you should know about bacteria reproduction, which also plays into their anatomy as well as their adaptations. They reproduce primarily through an asexual method called binary fission. And it's called asexual because there's only one parent. So all offspring are identical. They're clones of the parent. And this happens really fast. you don't need another parent and to carry out any stuff. They just divide themselves, basically. So get an idea of how fast they can reproduce. I'm sure you guys all know that pregnancy lasts 9 months, which is very painful in humans. But then E. coli, which are a model organism. So a model organism is basically just a really well-studied or studied a lot organism that can be grown fast, tested on, and applied to a variety of science theories and hypotheses and experiments. So E. coli is an example of a model organism, and it reproduces every 20 minutes under the ideal conditions. So that means that if there are not enough nutrients, it's not going to reproduce. But under ideal conditions, once every 20 minutes, which is insanely short, part of why they picked it to be a model organism. Yeah. Again, explained here. So endospores are an example of adaptation. So endospores are resistant cells that form when the bacteria cell lacks some essential nutrients. So essentially, the bacteria first, so let's say this is a bacteria cell, maybe like a flagella, and then a plasmid. So they would make a copy of the chromosomes, sorry, not plasmid, but like the chromosomes, the genes, so now there are 2, and then they would basically like starve itself of nutrients, and then so this part would all die, and then so they would burst essentially. So when it bursts, it releases this new endospore. And so this endospore, it's very durable. It's coated with a lot of material, and it can last harsh conditions. So it can live for centuries. That's the extent to which it can survive. It survives in boiling water, cold, acidic. And so when conditions are right, again, it will gradually become active and reproduce, and then so the bacteria still gets to spread its genes, which is the goal of every single organism on Earth. So this is an example of an endospore. You have your cell wall. As you can see, it's very thick, all of it. Cortex. And then in the actual core, you have your chromosomes. Yeah. Okay. So how diversity actually arises. So you've heard me talk about binary fission in the last slide, but didn't I say that this is basically cloning? So how do we get new kinds of bacteria? And as you guys probably know, bacteria are very prone to evolution and mutations. That's why when we get an antibiotic and it doesn't work, it's because the bacteria has already developed resistance. How can it do that when it only clones itself so that its genes are the same? So if one disease killed one bacteria, wouldn't they kill all of them because they all have the same genes? Well, this is because of something called mutations. And mutations happen when DNA doesn't work like it is supposed to. So for an example, if you have a DNA sequence that's like A, G, G, T, and this codes for the protein structurelase. Let's pretend that's a real protein. And then you get a mutation. So one of these genes turns into a seed. This creates a new protein. It's called protein resistase. And then you have this virus, angry looking thing coming around, killing everyone, but it's actually resistant. This new protein is resistant. So this new protein gets passed down. So that's why mutations are important. So even though mutation rates are incredibly rare or low, because obviously like your body isn't going to make that many mistakes, but it's still 1 in a million. But 1 in 10 million, but considering how many times E. coli undergoes reproduction, that's 2,000 mutations a day. So that's 2,000 chances to gain immunity, to gain a favorable trait. And that's how diversity and why we have different kinds of bacteria arises. So the more, I guess, safe way of going about all of this is through genetic recombination, which is combining DNA from 2 new sources, so you don't have to rely on your own body going through some weird mutations. It's not the same thing as sexual reproduction because that occurs on the basis of like fertilization of an egg and a sperm, but obviously there's no egg and sperm involved here when it's literally just one cell. There is a bug in my water. Okay, anyway, so there are 3 main types, these 3. Collectively, this is called horizontal gene transfer, which is the transfer of genes from one organism to another horizontally. Wow. So first type is transformation, and this is when the genotype or phenotype of an organism is altered by foreign DNA. So for example, if you have a blue bacteria, but then here comes gene A, and GA turns the blue bacteria into red. That's an example of the phenotype or outward appearance changing. And the genotype is just the gene itself. So you have this new gene. And so an example of this happening is when a harmless bacteria turns into a harmful bacteria when it gets a disease-causing strain of DNA from the environment. That's what pathogenic means. So the chromosome basically takes the gene or allele, which is a version of a gene, and replaces its own. So the second type is transduction, and this is when phages carry, I don't know if I'm pronouncing that right, Fungi? I'm gonna go with phages, but it might be Fungi, whatever one you want, I guess. Carry genes from one host cell to another. And so phages are, no, it should be Fungi. I don't know, anyway. So this refers to the term bacteriophage. Bacteriophage, which is basically a virus that infects a bacteria. And so because viruses are technically not alive and they can't reproduce on their own because they don't have DNA material, they can sometimes inject it into a host bacteria and that eats it up, which eats it and transforms itself. Finally, we have conjugation, which is more direct. And this is when 2 cells are temporarily joined through a bridge. And so this is called a mating bridge, also known as a pilus, a sex pilus, which passes genes. It's hollow so the genes can travel through one another and DNA passes directly. So who can tell me what kind of horizontal gene transfer is on the right diagram? So is this transformation, transduction, or conjugation? Okay. It is conjugation because you have this bridge thing here, which it looks like green corn, but yeah. Okay, so moving on to actual more bacteria stuff that has less to do with reproduction and more with morphology. So, okay, so we have something called the Graham stain, which is a laboratory technique developed by someone named Graham. You probably expected that, and it separates most types of bacteria into 2 groups. This word is key because it means that not all of the bacteria can be classified into these 2 groups, unlike with domains. So the first step is that bacterial cells are put on a microscope slide, and they're soaked in this violet dye. This should be pronounced dye or spelled dye, my bad. It's treated with iodine, which is a chemical. And then the second step is that it's washed with alcohol and counterstained with a red dye instead of violet. So these two contrast each other, and it's usually saffron and yeah. If you think saffron, which is red. So you have your first type, gram-negative bacteria. And so here the dye is washed out. And so because the violet dye is washed out, the violet dye is washed out, you get a pink and red color, which is the color of your chemical. And so this means that you have a very thin peptidoglycan layer, which, if you recall, is a component of the cell wall. And it's surrounded by the second outer membrane, and the space between the cell wall and membrane is called the periplasmic space. And during the space, many proteins important to digestion, transportation, and detecting chemical gradients which can be used for energy exist between these two layers, periplasmic space. And the other type, as you probably guessed, is gram-positive bacteria. And this is where the violet dye, not violet dye is retained, so it appears blue and purple. And so they have 5 times as much peptidoglycan in their cell walls as gram-negative bacteria. And so instead of having a periplasmic space, because again there's no space, you have this thick meshwork of like various proteins and carbohydrates and stuff that basically serves the same purpose as a periplasmic space. So as you can see on the right here, like the purple things, that's gram positive. And then the pink, reddish, that's gram negative. Yeah. Okay, so now another way we can classify bacteria is based on shape. So based on their outward appearance. So it's directly what the eye sees. So, or the eye sees under a microscope. There's no chemical treatment and stuff. So you have 3 basic shapes. You have the cocci, which is a sphere. So, okay. So Angelina just asked me how I'm doing the drawings. So basically, there's something called annotations on Zoom, which like I just started using today. Or you can just click annotations, and then you can just like draw like. Oh, are you asking me like how I'm doing this with a mouse or something? Hi. Wait, so you're asking me how I'm doing this with a mouse? Wait. What is Angelina asking? What's happening? I know how. Oh, okay. Yeah, but basically I'm using annotating, and I have a pen here, so it's not like with the mouse because that's harder. But yeah, so cocci bacteria, they either live singly or associate in 2D or 3D chains. Blocks, chains, plates, blocks are some of the examples of shapes of colonies that they would take part in. Bacillus means rod-shaped, and spirillum means corkscrew, so like a spiral. And so these two may form, may be single or live in clusters and chains. Some other examples of shapes include like really long filaments or like branched filaments, so like this. So again, when you see here, you have your cocci, which are spheres. Here are some examples. You have your bacilli, which are rods. They're long. This is circular. You have your, these are all basically spirillum. They're like twisty. You have these budding ones, some examples of more shapes. Weird bean, yeah. So, moving on to the next slide, we're finally going to be looking at the different kinds of bacteria. This part always gives me a headache, so just like try to bear with me, and you don't need to remember anything at all, but if you would like flashcards too, I can provide them. But yeah, so our first group, we have low GC, gram-positive. And so this is also known as, this group is also known as firmicutes. So they have a low GC to AC ratio of nucleotides in DNA. So DNA is made out of 4 nucleotides, G, C, A, T paired with each other. And these two pair. So this means that in DNA, this pair occurs more than this other pair. And so, however, this part of their name is misleading because they're not actually all gram-positives. There are some gram-negative or no cell wall at all, which is an instance where you couldn't use the gram stain test. Some of these have endospores. For example, you have Clostridium bacillus, which causes human anthrax. Also, Staphylococcus, as an example of these bacteria, causes a lot of things like digestive diseases. Mycoplasmas are the smallest known organisms alive, and they're also part of this group. They have no cell walls. So the contradicting group is high GC, so they have a high GC to AT ratio, also known as Actinobacteria. Some of them have branch filaments, just like fungi. So if you recall, fungi have something called hyphae, which are like plant roots. And so fungi reproduce by having like these spores at tips, which form into new ones. And so these Actinobacteria, some of them are like that. Some other examples are bacteria that cause tuberculosis. Also the human, tuberculosis is the oldest known human, the oldest known human pathogen falls in this group. Also streptomyces, which are a huge group of antibiotics, is what actinobacteria are used for. Hypothermophilic are another group. They are heat lovers. Hyper means like extreme or like greater thermo heat, philic lover. So for example, the bacteria Aquifix lives in volcanic vents and hot springs, and then Thermotoga, another group. Within this group, they live in oil reservoirs, which are extreme environments. So the example on the right is Actinobacteria. As you can see, you have all of these branches and stuff. It's pretty cool. All right, moving on to part 2. We have 2 more slides after this, so please bear with me. So you have Hadobacteria, which also like heat. Hado is the Greek god of the Hado refers to Hades, which is the Greek god of the underworld. Examples of bacteria in this clade would be Deinococcus, which is resistant to radiation, cold, hot, basically all extreme. Also Thermus aquaticus, which is also known as Taq polymerase. It's a DNA polymerase. So it helps DNA replication in something called PCR. So also let me know if my handwriting is terrible. Actually, I'm just gonna fix it myself. Okay, PCR copies DNA fast. And it's used in like genome replicating and stuff. It's genetic engineering, so it's really cool. Cyanobacteria are blue-green bacteria, also known as this, because they are photosynthetic. So they have chlorophyll, which helps them photosynthesize. They release O2, which means that they are the basis of something called the green revolution, which is the increase of oxygen in our atmosphere. They use something called a photosynthetic lamellar system to help them perform photosynthesis. And the reason why plants can actually perform photosynthesis is because they inducted these cyanobacteria into their cells. So they ate them, and when they ate them, the cyanobacteria allowed them to photosynthesize as well. So they became before plants. They're actually the oldest organisms in fossil evidence, if that's interesting at all. So they can either be single or colonial, and if they're colonial, they have 3 different kinds of cells. You have the vegetative, which performs photosynthesis. You have the spores, which are like this resting stage, and heterocytes, which focus on nitrogen fixation. So you have the nitrogen cycle in the atmosphere, and cyanobacteria help play a part in that. So finally, on the cell, you have on this slide, you have the spirochetes. So spirochetes are gram-negative motile-bacteria, which means they can move. Thumbs up. That's a terrible, oh my God, let me undo that. Okay. And so they have axial filaments, which is their main thing. These are flagella in periplastic states, which when you remember, as you remember, is so you have this, you have your cell wall, and then you have your second outer membrane. This would be the periplastic space, and then so they would have flagella in this. They have spiral, they have corkscrew-like movement. So these are the bacteria on the right. They can cause Lyme disease, syphilis, and some of them live in mud and water, or they're parasites. So they infect a host. Let me clear. All right, our final slide for the sorry. We have 2 more groups to get through. First group is Chlamydia. They are the smallest bacteria. They are parasites, and they are gram-negative. So characteristics. They used to think that they couldn't make ATP, which is cell energy. It's what our main source of energy is. But they actually use something called translocase, which takes ATP from their hosts in exchange for ADP. So ADP, okay, wait. So ATP equals adenosine triphosphate. ADP is diphosphate. So if you add ADP, sorry, this is D, plus P equals ATP, that's how you form ATP. But then they just don't add the P, they don't bother trying to get it, and they just switch out the ATP of their host. Again, they're parasites. They have to live off of a host who don't gain anything from them, but they leech off of them essentially. So you have your ATP and they switch. But they can make ATP, they just choose not to. They have a very unique life cycle made out of elementary and reticulate bodies. I'm not going to go too deep into that because not anything we need to know. They cause a lot of diseases, including trachoma, STDs, probably most notable, and then pneumonia. So probably our most, I guess not important, but diverse group of the day is Proteobacteria. They are the largest group of bacteria, and they include all sorts of diverse members. You have your gram-negative photoautotrophs, which photosynthesize. They can also metabolize sulfur, which is something that not a lot of organisms can do. Bacteria of this group are believed to have formed mitochondria. So if you think cyanobacteria gave rise to a photosynthesis, then Proteobacteria gave rise to cellular respiration, which makes all of our energy. Also includes the nitrogen-fixing rhizobium, which help, so you have your plants, and then you have your plant roots. And so in these roots, you're gonna have like bacteria that help fix nitrogen for it. Nitrogen helps it grow. It is essential for growth. E. coli is also part of Proteobacteria, also different bacteria that cause the plague, cholera, salmonella, crown gall. But there's also good ones like Fimbriae, which is a bioluminescent genetic marker, and bacteria in yogurt. So they're all good. All right.